Okay, guys, this is part 2 of our chapter 3 really long discussion of cells. This part's going to be dealing with cellular structures and functions. Previously, we talked about cellular transport. So the cytoplasm is kind of encasing all of our cellular organelles. So it's the material that's located between our plasma membrane, which we just talked a lot about, and the nucleus, which is the control center, the brain of your cell. So the cytoplasm is composed of cytosol, inclusions, and organelles. So the cytosol is the actual, like the gel that you're used to thinking about for the cytoplasm. It's made up of water, some other molecules like protein, salts, sugars, things like that. Our inclusions are insoluble molecules, so they're not going to dissolve in this. This can vary with our different types of cells that we have in our bodies. We talked about how many different types of cells and things like that we have back in the first part of this. And then our organelles, of course, these are all of the little pieces of machinery that are found inside of all of our cells, which can also differ based on the different cell type. And these are going to be like small little organs that all play like an essential role in order to keep all of our cells functioning correctly. They are all, well, not all, most of them are contained in membranes, and we're going to talk a little bit about cellular organization and how that affects our organelles in just a minute. So we have membrane-bound organelles, which you've heard me talk about, especially if you were in my freshman class, and we have non-membrane-bound organelles or non-membranous organelles. So membrane-bound organelles are organelles that are surrounded by a lipid layer or a lipid bilayer, depending on the different type of organelle specifically. So this would be like our mitochondria, our endoplasmic reticulum, our Golgi apparatus, peroxisomes, lysosomes. Okay, our non-membranous organelles are going to be our ribosomes, the cytoskeleton, and our centrioles. This just means that these three organelles do not have a lipid layer around the outside of them. I like to think about our eukaryotic cells because this is anatomy and physiology. We are humans. We have eukaryotic cells inside of our bodies. We have our very highly organized, compartmentalized cells, these eukaryotic cells, right? So membranes kind of act like, I have an analogy with a refrigerator, right? Your refrigerator. You put your food in your refrigerator. If you buy milk from the grocery store, it comes in a carton, and you put the carton inside of your refrigerator, and it doesn't touch anything else. I mean, the carton might, but the milk is not spilling out all over the place. You can have your milk and your orange juice right next to each other, but they're not going to mix, right? And if you do mix them, that's going to be pretty disgusting. And that's the same thing that happens inside of our cells with our organelles. If you mix them together, it can be kind of disastrous. Not that it would taste disgusting, but you could have your lysosomes mixing with something else, which means that it's going to destroy whatever else it's mixed with. So we have this compartmentalization, and I like to think about it, like I said, as like containers around food in a refrigerator. It is highly organized. Everything is separate. So that's one way that you can kind of imagine our cells are built inside of your mind. So our mitochondria is the powerhouse of the cell. Why is it the powerhouse of the cell? That's like the one thing you need to know about biology. If you remember anything, mitochondria is the powerhouse of the cell. Congratulations. Why? It's the powerhouse of the cell because it's manufacturing ATP, adenosine triphosphate, which is a compound that holds our energy, and we can lyse off the terminal phosphates and create ADP and then eventually AMP, and we can release a bunch of energy to help drive chemical reactions. So it's also called a power plant, the powerhouse of the cell. Our mitochondria are enclosed by double membranes. We have an inner membrane that has a lot of folds. These are called cristae. They're embedded with different membrane proteins that are going to help with cellular respiration, which we'll actually talk about in our next unit. Mitochondria are actually pretty cool because they have their own DNA and RNA, as well as ribosomes. And this is where our endosymbiotic theory comes from, that the mitochondria and chloroplasts were once their own organisms. So they were once their own cells, and then they were just engulfed by these larger cells, and that they were living in, endosymbiosis, essentially. So we have one living inside of the other, and some evidence here is that it has its own ribosomes and DNA, which is pretty neat. And our mitochondria also closely resemble bacteria, just based on doing fission. So this is like binary fission. They get a little bit bigger and then they can divide. Bacteria do the same thing. Pretty neat. Here's an example of what a mitochondria looks like. So you see that it has a double membrane there, the outer membrane, and then you also have the inner membrane, which makes all those little cristae, all those little lobulated folds. And then you can see the enzymes that are located on the inner mitochondrial membrane. And like I said, all of those are involved in cellular respiration, which is something that occurs in all organisms. Some of them just use aerobic versus anaerobic. So aerobic respiration using oxygen would take place inside of the mitochondria. Next, we have ribosomes. You know that ribosomes make proteins. Okay, so they are non-membranous organelles. They are actually two separate subunits. I draw them like a Krabby Patty. It's like a top bun and a bottom bun. The top bun's a little bit bigger. It's a large subunit and a small subunit, and they're actually separate unless there is an mRNA present for them to attach to, bind to. So we have, this is a non-membrane bound organelle because it actually exists as two separate pieces until there's mRNA for it to work with. So it's made up of protein and rRNA. The rRNA stands for ribosomal RNA. So literally it's just this like blob of protein and rRNA. And we have two different forms. We have the free-floating ribosomes, which are floating around the cytosol and cytoplasm. And we also have membrane-bound ribosomes, which are attached to the ER, and that's why it's called the rough ER, because they're actually, like, it's dotted with a whole bunch of our ribosomes. So those are the differences between those two there. The endoplasmic reticulum itself, like I said, you have rough ER, you also have smooth ER. So it's a lot of interconnected cisterns. It's kind of like those like lobulated, like squiggly looking guys again. It's flattened membranous tubes that are full of fluid, and the ER is continuous with the nuclear membrane. So it's actually like just a continuation of the nuclear membrane. And like I said, we have our rough and our smooth ERs. Those are the two separate types. The rough ER has the bound ribosomes, whereas the smooth ER does not. Here's a picture of what that looks like for you. So as I said, it's a continuation of the nuclear membrane. And then you can see where you have the smooth ER, there are no little dots or ribosomes on it. Whereas if you have the rough ER, that has a bunch of dots, which are ribosomes attached to it. So the rough ER, like I said, this is where we have our ribosomes attached. So it's the site of synthesis of proteins that are going to be secreted from the cell. We also make proteins like that are going to be present in the plasma membrane, so not necessarily like secreted for like release, but they're just secreted to the other side of our cell membrane. So we also have proteins that are going to enter the cisterns as they're synthesized and then be modified as they are winding through all of those fluid-filled tubes and passages. And then further, they can be edited in the Golgi apparatus in order to have like these little tags, like certain glycolipids or glycoproteins attached to the outside of them that kind of signifies like where they're supposed to go in the cell. It's like marking them for transport. That happens in the Golgi apparatus. So alternatively, we have the rough ER analysis of the smooth ER. It's a network of looped tubules that are continuous with the rough ER, so it's all attached together. And again, the ER is continuous with the nuclear membrane. We have enzymes that are found in this plasma membrane to function doing like the following: lipid metabolism. We have absorption of some of these lipids. We have detoxification happening here. So you'd have a lot of smooth ER and maybe like your liver, maybe your kidneys. You have the conversion of glycogen to free glucose happening here, storage and release of calcium, and other some other functions as well. A lot of them are dealing with lipid metabolism, though. This is where a lot of this is actually occurring. Next, we have the Golgi apparatus. The Golgi apparatus is, again, like a membranous, long, lobulated, flowing structure that we have inside of our cells. So it's a stacked and flattened membranous cistern of stacks. So it looks similar to the ER, it just doesn't have those bound ribosomes on it. It's going to modify our proteins and lipids to help signify where they're going to go. So the rough ER with those bound ribosomes is going to be the site of where we're going to create our proteins, maybe start the modification process of proteins. And and then we're going to finish that process in the Golgi apparatus. So there's a couple steps involved. We have transport vesicles from the endoplasmic reticulum that are going to fuse with the interface called the cis face of the Golgi. Then we have our proteins and lipids that are going to be taken inside of our Golgi apparatus to be modified and, like I said, like sorted and tagged. So they're kind of like getting these like extra like receptors that are going to signal where they're supposed to go inside of the cell or outside of the cell. And then the Golgi is going to act as a traffic director to control different pathways that these products, these end products, are going to take as they're leaving off of the trans or the outer face of the Golgi apparatus itself. So this is kind of what that looks like. Like I said, it's this long kind of blobby, lobulated structure here. So we have vesicle that's coming from the rough ER. So this is on the cis face, the internal face. So that's the part closest to the nucleus. We're going to receive the contents, could be proteins, could be lipids. We're going to alter them in whichever way that particular protein or lipid needs to be altered. And then you will have on the trans face or the shipping side of the Golgi apparatus, you will have a vesicle butt off from there. And then it will deliver these materials where they're supposed to go. So depending on the different types of contents that we have, the final transport vesicle can take certain paths. So we have pathway A, B, and C. If it's a secretory vesicle, it's going to be containing proteins that need to be on the outside cell, the outside of the cell, or just secreted in general. So we are going to have that secretory vesicle is going to fuse with the cell membrane and have exocytosis. Man, words are difficult today. Exocytosis will occur in order to release these products to the exterior surface of the cell. In pathway B, we have the vesicles containing lipids or the transmembrane proteins fuse with the plasma membrane or the organelle membrane if it's supposed to be destined to a different organelle. And then we're going to have the contents inserted directly to the destination membrane. So if we have like proteins that are supposed to be like transmembrane proteins that are going to be exported to the cell membrane, then they will fuse with it and then just stay right there. The whole vesicle will actually become part of that organelle. In this case, in my example, the plasma membrane. In pathway C, we have lysosomes that are containing digestive enzymes, so hydrolytic enzymes. They remain in the cell holding the contents in the vesicle until they're needed. So we just kind of have some extra vesicles that are kind of hanging around with some contents that we might need, we might not need. If we don't need them later, then they'll fuse with the lysosomes that have those hydrolytic enzymes to be digested later. So here's another example of what we just talked about. You have the proteins and lipids entering into the cis face of our Golgi. You have them going through to be modified and however they're supposed to be modified. And then you have them budding off of the trans face and then going to their specific locations. So you see pathway A, we have the vesicle contents destined for exocytosis. You have pathway B, the vesicle membrane, to be incorporated to the plasma membrane, as I said. And then in pathway C, the lysosome containing hydrolytic enzymes is going to help to digest any extra materials that we have once they're no longer needed. Next, we have peroxisomes. So our peroxisomes are pretty cool. They help to detoxify our cells. We're actually going to do a lab on this. It's a pretty neat lab of how this happens in our bodies. So we have these like sacs that have these detoxifying substances that help to neutralize toxins. So we have these things called free radicals that we're exposed to all the time and can get into our body. They're toxic. They're highly reactive molecules. They're natural byproducts of our cellular metabolism as well. So they can definitely cause some damage if we don't take care of them. So that's what our peroxisomes are built for. It's an area where we can have a high concentration of oxidases. So oxidases are going to use oxygen to convert these toxins into hydrogen peroxide. To break down this hydrogen peroxide. No, we don't want hydrogen peroxide. That's a bad thing for our cells. So we have hydrogen peroxide, H2O2, and we're going to break that down into two completely harmless molecules called water and oxygen. So it's pretty cool that we have all these oxidases that are present inside of the peroxisomes that are able to detoxify these harsh chemicals into things that we can actually utilize, water and oxygen. These are both things that our body needs, and we're able to break down these harmful substances into harmless substances. We talked about lysosomes earlier, so remember that these are sacs that contain hydrolytic enzymes. acidic hydrolases or acid hydrolases, they are going to be breaking down whatever is contained in a vesicle that the lysosome fuses with. So it's used in metabolism to break down things. We sometimes break down like glycogen. We break down other large molecules. We break down things that are brought into the cell through endocytosis that could be potentially harmful, that are kept in a vesicle. So this is just cellular digestion. This is kind of like the stomach of the cell. And if we have this released into the cytoplasm, you can imagine that all these, hydrolytic enzymes whose job it is to literally break down everything that they come in contact with, it's not going to be a very good thing for the cell if these are going to bust open or, if the cell triggers some sort of release. This is an autolysis, so the cell would be digesting itself. And this happens in some cases where you might have a cell that's infected with a pathogen or something, then this is part of the immune system that can actually trigger this to help kill that particular cell. That way we don't have things spreading, which is nice. So here's an example of what your lysosomes would look like. You have the lysosomes are these orange blobbies, and then we have light green areas of material being digested within them. Next, we have an endomembrane system. So this is our membranous organelles that we've discussed so far, like the endoplasmic reticulum, like the Golgi apparatus, the secretory vesicles and lysosomes, as well as the nuclear and plasma membrane. So these are our membrane-bound organelles. These membranes and organelles work together in order to produce, to degrade, to store, to export biological molecules, such as our lipids, our proteins, right? And then they also degrade potentially harmful substances. We talked about like the lysosomes are helping us to protect ourselves in case like we get infected or our cells are getting infected, or having a surplus of extra materials in the cell can be harmful, or waste that needs to be disposed of can be harmful as well. So we have things like our peroxisomes and our lysosomes that help us take care of those problems. So here's a picture of like what we're talking about, right? We have our endomembrane system here. You have a eukaryotic cell. This is particularly an animal cell. I know this because it has a nucleus, which tells me it is eukaryotic, and we do not have any chloroplast or a large central vacuole or a cell wall. Therefore, I know that it is a animal cell. It is an animal cell, and this is highlighting all of the organelles that are part of the endomembrane system, which is a lot of continuity here. You have a lot of things that are very continuous, like the nuclear envelope. You have the endoplasmic reticulum, the Golgi apparatus. They all kind of like lobulate out from our central nucleus, and then we have some of the other things that we talked about with our lysosomes, vesicles, things like that. Next, we have like another network. So the endomembrane system is like a whole network. Cytoskeletal system is also like a network of things working together. So it's an elaborate network of rods that run throughout the cytosol. So remember, that's like the jelly stuff between all of the organelles. There's hundreds of different kinds of proteins that link these rods to other cells. We have the linking of organelles to these different fibers. These fibers can help to hold cells together. These cells, or these parts of the cells, can help to mobilize different organelles within the cell. They have a lot of different functions. They also act as the cell's like bones, ligaments, muscles, and playing a role to help things move around, like I said. So there's three different types of fibers that we're going to be talking about: microfilaments, intermediate filaments, and microtubules. Those are the three things that make up our cytoskeleton in our cells. So our microfilaments are the thinnest of all the cytoskeletal elements. They are sometimes referred to as like actin filaments. So we have our microfilaments. They are semi-flexible strands of protein called actin. So they are also called actin filaments. Same thing, microfilament, actin filament. Each cell has a unique arrangement of strands, although they share a common terminal web. So we have a dense cross-linked network of microfilaments attached to the cytoplasmic side of the cell membrane. So that's the interior surface of the cell membrane, and it helps to strengthen the cell surface and helps to resist compression. So while we don't have cell walls, we do have structures to help maintain the integrity of our cells. Yes, our membranes are flexible and what I refer to as, quote, squishy, but we do have these cytoskeletal components to help give our cells structure to prevent them from being crushed, to help maintain their structure. And some of these microfilaments are also involved in cell motility and other, organelles and things that are moving around inside of them, and changes in the cell shape, such as endo and exocytosis. We're absolutely moving the cell, right? Because we either have things entering or exiting in these vesicles that are fusing with the cell membrane. So we'd obviously have a change in the cell shape temporarily, at least. So here's kind of what an actin subunit would look like here. You see that it's kind of this like bat wing looking blue filament in this picture. The next one is going to be our intermediate filaments. So these are intermediate. If our microfilaments are the smallest, these are our intermediate sized. So it's between a microfilament and a microtubule. So these are tough, insoluble, rope-like protein fibers. They're composed of a tetramer of fibrils, which are twisted together, which we'll look at in a second, resulting in one stronger fiber. They help the cell resist pulling forces. So the filaments attach to desmosomes, the desmosome plaques that are on the interior side of our cells, and act as internal guy wires. So essentially, they're helping to, protect the cell from, if you're linking them together through the desmosome plaques, then that you have other fibers that are going to help attach cells together. But this helps to resist pulling forces. So like if there's a lot of pressure on one cell, the desmosomes are supposed to act like Velcro, that you're not going to like break the cell because you have a whole bunch of tension, that they'll just separate and then they can later come back together. Some have special names. We have like the neurofilaments in nerves, keratin filaments in our epithelial cells. So sometimes they have particular names that reflect where they're located, what they're composed of, or what they're associated with. So here's an example of what that looks like. So you have these like tetramer subunits that are all kind of like woven together. You can tell that this is a 10 nanometer thick molecule, whereas our microfilaments were 7 nanometers. And they kind of form this, in this picture here, this like purpley lavender network that's surrounding the nucleus of this cell. Lastly, we have our microtubules. You've probably heard about these before. So they're the largest of the cytoskeletal elements. So they consist of hollow tubes composed of protein subunits called tubulins. And these are being assembled and disassembled constantly. A lot of them come from centrosomes, which is like a central area of the cell. This is actually where the centrioles develop when we are going through like mitosis, and we develop microtubules called spindle fibers to help pull the chromosomes apart. So the centrosome is kind of like the area that these like radiate from. So they help to determine the overall shape of the cell and the distribution of the organelles, because this is actually how we move organelles around. So many organelles are tethered to microtubules to keep the organelles in place. So they keep everything in close association with whatever other organelles they're supposed to be close to according to that particular cell type. We can also move substances throughout the whole cell with the help of motor proteins that are actually going to they have these like little subunits that look like tiny, tiny little fat legs that are going to literally walk one foot in front of the other kind of along these microtubules, using them as like a road. So we have these tubulin subunits here. In this picture, we have the like golden orangey colored network that's surrounding the nuclei in this cell. So again, these are our hollow tubes. These are the largest of our cytoskeletal elements that help support the cell and to help generate movement of our organelles around the cell. So these motor proteins that I talked about, they're complexes that function in motility, so they can help in movement of organelles and other substances around the cell. They're using, like I said, our microtubules as like a track or like a road to move whatever substance they're carrying, and they're powered by ATP. So this would be like definitely more of an active than a passive sort of process happening. So we have centrosomes and centrioles that I mentioned earlier. So the centrosome is located near the nucleus, and it means the cell center, like central, right, center. These all kind of have the same prefix. So it's a microtubule organizing center, and it's going to have these centrioles, which are a pair of barrel-shaped microtubular organelles that lie at right angles to each other that are within this centrosome. And this is going to help, like, this is where the spindle apparatus develops. So, like I said, for cellular division, this will help pull the chromosomes apart and things like that. And then our centrioles also form the basis of our cilia and flagella that are used for cell mobility. So, the flagella is typically a whip-like tail that extends from the cell, and our cilia are these little tiny finger-like structures that kind of like wave in order to help with cell movement. So here's what our centrioles look like. You can tell that you have these hollow tubulin structures that are all present within the centrosome region. And then you have the microtubules that are going to be further extending from that in order to help organelle movement. And like I said, like help us during cellular division. Here's another picture that you see these like triplets. That's typically how microtubules are arranged, and you can tell that they are hollow. So as we talked about, our cilia and flagella, they are on the exterior surface. So we have certain cells that have structures that extend from the cell itself on the surface that help with like movement, like our cilia and flagella aid in the movement of cells or of materials across the cell surface. So like cilia are often present in areas where you're needing to sweep or move things along, like mucus, things like that, like in your intestinal tract. Like we have particular ones called microvilli that are finger-like projections that extend from the surface of the cell to increase surface area. This helps with our absorption of nutrients and water, and also they're mobile. They are moving a little bit to help move things along your intestinal tract. So cilia are these little tiny whip-like modal extensions on the surface of our cells, such as our respiratory cells, because they are constantly like sweeping. And like I said, mucus, I mentioned that just a minute ago. Flagella are much longer, and they're there to help to actually drive motion of the entire cell, not just something past the cell like a fluid. They're actually causing the whole cell to move. So they're a lot longer. For an example, here you have like the tail of a sperm cell. Both of the structures are made up of microtubules that are synthesized by these centrioles. Remember that those are the little orange guys at right angles to each other, yellow orange guys that are located at right angles to each other inside of the centrosome that are called the basal bodies because they form the base of the cilia and the flagella. So, or cilium is one and flagellum is one. So the cilia and flagella have a 9+2 rule or pattern. So this means that you have 9 sets of double tubes surrounding a central pair of doublets. I'm not so concerned about this rule. You just need to know that obviously it's microtubules that compose these things and help to create the basal body that stems from the centriole. Cilia movements alternate between power stroke and recovery stroke. This alteration produces a current at the cell surface that moves substances forward. So this is how cilia actually function. You're more familiar with like the spiral or whipping motion of a flagella as being this, long tail that's actually driving the whole cell to move, whereas the cilia are going to be alternating between these power strokes and recovery strokes in order to move substances past the cell. And when you have a whole bunch of them working together in a tissue, you can sweep substances along the surface of the cells pretty rapidly. This is just more of like the 9+2 rule, the total composition of these different structures, both on the internal and external surface of our cells. And this is for the cilia. And like I said, this layer of mucus, this is helping to move it past these cells. The microvilli that I talked about, that are increasing the surface area of your intestines. They're these tiny, tiny little finger-like extensions of the plasma membrane that project from the surface of select cells, like our intestinal cells, our kidney tubule cells. They increase the surface area for absorption, which I mentioned again, and they have a core of actin microfilaments that's used to stiffen the projections to make them have a little bit more of a rigid structure. And this is what they look like. So these are smaller than our cilia and our flagella, and they're there for increased surface area to help increase the rate of absorption of nutrients and water. So we have our nucleus. Our nucleus is the most famous of our organelles. It is the brain of our cell. We have the powerhouse, which is the mitochondria. This is the brain of the cell. This is where our DNA is located, is the genetic library. This is very important. It's the largest organelle. It contains your DNA, which is important for synthesizing your proteins and basically, making you who you are. It's pretty important. We have some cells here that are, most cells that are uninucleated, so they have one nucleus. We have a couple that are multinucleated, such as like liver cells, and red blood cells actually do not contain a nucleus. They are anucleate. They do not have one. Actually, in the center of our red blood cells, you have like a little dip. It actually gets like thinner there, and that's where the iron and heme groups are, which are going to attract oxygen to them. So when it, you have oxygen coming into your lungs and it's diffusing through these little capillaries, they're actually able to grab onto the oxygen that way, where they wouldn't be able to. The structure would be a little bit different if they had a big old nucleus in the middle of them, things like that. Remember, structure dictates function. Within our nucleus here, we have a nuclear envelope, which envelopes, the nucleus. So it's around the outside, like an envelope is around the outside of a letter. The nucleoli, nucleolus is going to be within that. And then our chromatin is referring to a structure that our DNA is capable of making. So here we see a picture of the nucleus. You can see the little pores that are present in the nuclear envelope. The whole nucleus itself is just that purple region. You have the rough ER, which again is an extension of our nuclear envelope. So the nuclear envelope is a double membrane barrier that encloses the nucleoplasm, or that's basically the center of the nucleus. So the outer layer is continuous with the rough ER, like we've been talking about. The inner layer is called the nuclear lamina. It's a network of proteins that help to maintain the shape of the nucleus and acts as a scaffolding for DNA. We also have these things called nuclear pores, just like your face has pores, your skin has pores. The nucleus has pores to allow substances to pass in and out of the nucleus. They're guarded by the nuclear pore complexes, which help to regulate what things can come in and out. So like, for instance, we're going to obviously want to release mRNA because DNA is stuck inside of our cell nucleus, and we need to get that message out to the ribosomes in order to make our proteins. So we're going to use nuclear pores to release that mRNA to the cytosol, so then we can get that mRNA over to a ribosome to create a protein. Here's some pictures of our nucleus. You can see the little dots are the nuclear pores, things like that. So the nucleoli, these are the dark bodies within the nucleus that are involved in creating rRNA, which is ribosomal RNA. So rRNA in a protein, remember that's what's going, and different proteins are going to make our ribosomes. So this is essentially where our, quote, ribosomes are created. We're just creating the rRNA inside of the nucleolus, which is inside of our nucleus. Next, we have chromatin. So like I said, this is a structure our DNA is capable of making. So it's 30% thread-like strands of DNA and 60% histone proteins and 10% RNA. Histone proteins are proteins that help to tightly coil the DNA. Because of all of the charges extending from DNA, it's negatively charged. As it's coiling on itself, the same charges don't like to stick together, opposites attract. So these histones help to alleviate some of that tension of all these negative charges compressing on each other. So that's what's it helps the DNA wind up. So the chromatin is arranged in fundamental units called nucleosomes, which consist of DNA wrapped around histones, which I just talked about. You have chemical alterations of histones that have an effect on the DNA and can help to regulate gene expression. So sometimes you can add different groups to our histones to help the DNA pop off of the histone temporarily, which means that you can access that part of the DNA because it's not tightly wrapped around something anymore, which means those genes are going to be expressed more frequently than if you have DNA that's tightly bound to a histone. That's all part of gene regulation. Recall that chromosomes are just our condensed chromatin. So our chromosomes is like X-shaped things that you're used to looking at. So the condensed state helps to protect fragile chromatin threads during cellular division. So chromosomes are highly organized chromatin. So here's an example of kind of what that looks like. You have your, a chromatid is 1/2 of a chromosome. You have all of the chromatin that's supercoiled to create our chromosomes. Again, this is more pictures of our nucleus that we talked about. This is, of course, where our DNA is located inside of the nucleus because we have eukaryotic cells. The next topic is going to be the cell cycle. So this is going to be a pretty brief section. So we're talking about like mitosis here. So PMAT, PMAT, prophase, metaphase, anaphase, telophase, and then of course cytokinesis. That is mitosis. The cell cycle is the entire event of both cell growth and doing its daily functions called interphase, followed by mitosis, which is a very quick 10% of the cell's life of when it's actually dividing. So the cell cycle is a series of changes a cell undergoes from the time it's formed until it reproduces. There's two major phases that I just mentioned. Interphase, which is when the cell grows and carries out its usual activities, and then the cellular division or the mitotic phase, which is when the cell actually divides into two. So this is the first time when division and multiplication are the same thing. Pretty neat. So in interphase, this is where the cell spends about 90% of its life. It's a period from the cell formation to cell division. So in the meantime, from creation to when it's going to divide again, this is when the cell is going to be carrying out, it's going to be growing, it's going to be carrying out whatever purpose it's made to do. If it's a red blood cell, it's going to be doing its thing, carrying around oxygen, removing carbon dioxide from all the tissues in the body. So during interphase, we have the nuclear material in an uncondensed chromatin state, because we're not worried about dividing yet. So it hasn't been like super coiled up yet. Interphase consists of subphases that we're going to talk about, including DNA replication. So these are the subphases, G1, S, and G2. G1, called gap 1, is when we have growth and metabolism. So this is when the cell is doing its daily jobs, daily functions. This is what it's supposed to be doing. This is why we have this particular cell. Cells that permanently cease dividing are said to be in the G0 phase. It says G0, but it's NAUGHT, I believe, or I think. G0 phase. I'm a scientist, not an English person. I apologize for my spelling. Right, this is G0. So this is when we have cells that are not dividing anymore. This might be because your body signaled that there's something wrong with the cell. It's going to go into the G0 phase because it could potentially be cancerous and we don't want that to grow anymore. It could be something like when you get old, like Miss Huber, you have nerve cells that's like stop dividing. They slow down division and eventually stop dividing in your lifetime. After this G1 gap phase where we have growth and daily functions, metabolism, we have something called the synthesis or synthetic phase, which is when DNA is going to be doubled, right? The whole goal of mitosis, T for 2, we're creating 2 cells. You need 2 sets of DNA because without the DNA, the cell has no idea what to do. So the synthesis phase is when we're going to double that DNA. So normally our cells have 46 chromosomes. During the S phase, they're going to have 92 chromosomes, twice the amount of DNA, because then when we divide, it'll be restored 46 and 46 and everybody is happy and healthy. That's how it works. So after the S phase, we go through a phase called G2, which is preparation for division. The cell is going to start develop the spindle apparatus and things here in order to help us organize all of our DNA during actual mitosis, which is cellular division. Here's a picture for you to kind of show you what's going on here. So interphase is this like green teal part. You see it's huge. It's huge compared to the mitotic or the M phase because that's where the cell spends 90% of its life. The mitotic phase, the division, is just this quick little tiny part, but it spends most of its life growing and doing its job, and then it's going to double its DNA because it needs 2 sets of DNA to make 2 cells that are healthy. And then during G2, it's going to be like, all right, we're about to divide. Let's do our final preparations. Let's make sure that that DNA has doubled correctly at the G2 checkpoint. Then we're actually going to divide prophase, metaphase, anaphase, telophase, and cytokinesis. That's how the whole cell cycle works. So the cell cycle, 2 words, 2 phases, interphase and M phase. You probably are thinking, oh no, it's more than that because it's got PMAT. No, the whole cell cycle is just interphase and mitosis. There's 2 phases happening there. Now mitosis has more steps, which we'll talk about in a minute. So during S phase, we have DNA replication. So prior to division, the cell is going to make a copy of the DNA. Like I said, you have to have 2 cells at the end of this. You need 2 sets of the DNA. You have the double-stranded helix unwinding, unzipping, and you're going to use helicase and topoisomerase to help with all of that. Those are enzymes to help with DNA replication. You have a replication fork, which is the point where the strands are separating. You have the replication bubble, which is the area where the replication is actively going under replication. where the DNA is going under replication. You have one half of the DNA acting as a template strand. Remember that we have a semi-conservative model of DNA. Half is old, half is new, half is red, half is blue. Typically, those are the colors that are used to signify new and old DNA. An RNA starts off the replication by laying down a primer to kind of be like, hey, all these enzymes, you need to pay attention here. This is where we're going to start. So we have a bunch of players involved here and to help out in this process. DNA polymerase is going to help to attach the primer and begins adding attached to the primer, and it begins adding nucleotides to form the new strand. Because remember that you have one strand that each of those little nucleotides needs a buddy. A pairs with T, G pairs with C. Remember that you also have a leading strand and a lagging strand because DNA is anti-parallel. So one side is from 3' to 5', the other is from 5' to 3', and you have your leading and lagging strands because the DNA is going in opposite directions. Our DNA polymerase only likes to add to, one particular end of our strand. So it only moves in one direction. And we'll definitely look at pictures because this is a lot easier if you have something to look at. DNA polymerase, like I said, works in only one direction. It cannot go, quote, backwards. So we have to have a whole bunch of extra primers, those RNA primers on our lagging strand. Think about video games. Lag is not a good thing. It means that it's all broken up and choppy and awful, and you run into a wall forever and look stupid. That's what's happening with the lagging strand. Okay, you have all these little tiny segments called Okazaki fragments that you have to stitch back together with DNA ligase. So it's a much more complicated process than the leading strand. In the end, our leading and lagging strands are going to create our two identical daughter DNA molecules because you need two sets of DNA because you're going to make two cells. Okay, so like I said, we use the semi-conservative model for our DNA replication. Half is old, half is new, half is red, half is blue. And we will look at that further in a little bit. Here's the overall process of DNA replication. Notice the lagging strand goes in one direction here, whereas the leading strand goes in the opposite. The lagging strand is the one that's made in little segments called Okazaki fragments, whereas the leading strand is synthesized in one complete continuous strand. You see all the little free nucleotides being added in there by DNA polymerase. You also see that you have a helicase, which is unwinding and unzipping our DNA. And here we will create two genetically identical copies of this DNA. So most cells need to replicate continuously for growth and repair purposes. That's the whole point of cellular division. Why do we need to make more cells? Because we got to grow and we got to fix ourselves. So we grow. You are not the same size today as the day that you were born. Thank goodness for your mother. You were once just one single zygote cell. You were one single cell and now you are trillions of cells. You definitely have grown. Okay, repair purposes. You don't stay injured forever, except in a couple cases, right? But like, let's say that you get a scab because you fell down on the playground when you were a kid. Your knee is still not scabbed. You might have a scar if it was really bad, but you're not still openly bleeding all over the place from an injury that happened a while ago because of cellular division, right? We have our skeletal, cardiac, and nerve cells that don't divide very efficiently, and those damaged cells are often replaced with scar tissue. It's just a more difficult process for those particular types of cells, and they divide a lot slower. The M phase of the cell cycle is the phase where the actual division occurs. You have two different events. You have mitosis and cytokinesis, which a lot of books say that cytokinesis happens at the same time as telophase. It just depends on the adaptation that you're looking at. We also have control of cellular division, because control of cellular division is important because when you have uncontrolled growth of cells, it's called, cancer. So it's really crucial to control when cells are going to divide. So this is part of that cellular communication that we talked about a little bit previously. You can't have all your cells divide at the same time, or none of them are doing their job and then you're going to die. So it's very important to control the overall process of cellular division and which cells are doing it at what times and when each one is doing it. So again, our overall process, interphase, mitosis. Okay, mitosis is just 10% of the cell's life, and it is mitosis followed by cytokinesis, or like I said in some textbooks, it says that telophase happens at the same time as cytokinesis. So we have mitosis, which is the division of the nucleus, whereas cytokinesis is the division of the cytoplasm. Okay, so here we have four main stages: prophase, metaphase, anaphase, and telophase, affectionately called PMAT. If you can't remember the order, it is PMAT, PMAT. Prophase, metaphase, anaphase, and telophase. Of course, we're going to be doing our mitosis hand model later. Prophase is the very first one. Okay, so this is when we have chromatin condensing. We're going to form visible chromosomes, so everything is packed super, super tightly together. Each chromosome and its duplicate are held together by a centromere. The centromere is going to be important for our spindle fibers to attach to. So the mitotic spindle, that's what we're kind of like referring to here. The spindle apparatus, mitotic spindle is the same thing. So each of these little microtubules that we talked about previously is going to actually attach to the kinetochore and the centromere. That's going to help to equally split these chromosomes later when we actually get to the division part of this. In late prophase, the nuclear envelope starts to break up. So in your textbook, you'll see it as maybe like a dashed line or a dotted line. Special microtubules attached to specific areas on the centromeres called kinetochores. And like I said, this is going to be to pull them apart later. So here's some examples of what that looks like. You have your spindle fibers, you have the centromere, and the chromosomes are tightly compact, and they have two sister chromatids. We have our dissolution, the dissolving of our nuclear membrane here in order to open up everything and allow everything to kind of like start to attach to the kinetochores and the centromeres in order to start pulling the chromosomes into, a line for the next phase. That's metaphase in the middle. We're going to have all of our DNA line up in the middle plate, the metaphase plate, along the cell's equator, just like this. So the spindles have pushed and pulled all of these chromosomes into a straight line because we're about to split the cell, so we need them along the equator, so then we can separate everything evenly and make sure that everything is divided correctly. Anaphase is the shortest of all phases. It's when we have the actual splitting of our chromosomes. So the centromeres of the chromosomes split simultaneously. So each sister chromatid now becomes a separate chromosome. Okay, so you have like those little X shapes. Okay, we're gonna have that open up. All right. Here's what that looks like. So we have our own chromosomes now. We have these sister chromatids attached together. Now we're separating them. We're pulling them to the opposite poles. Why? Because that's where the new nuclei are about to form in telophase. In telophase, it begins when we have the chromosome movement stopping. It's going to be at both poles, the extremes of our elongated cell here. And each set of chromosomes, they're going to actually uncoil to form chromatin again, because remember that we only have our chromosomes when we're actually dealing with the division of the nucleus, or the division of the DNA, which has, happened now. And we have new nuclear membranes starting to form around this chromatin mass. We start to see the nucleoli or the nucleolus reappear, and the spindles are going to disappear because we don't need them anymore. They're not there to push and pull around all of our chromosomes anymore. They're already where they're supposed to be, and they're already in these newly forming nuclei inside of the cell. So we have two nuclei in the same cell. Telophase, two nuclei in the same cell. Cytokinesis, so here it's saying that it begins in late anaphase and continues through mitosis. Some textbooks will tell you it starts in telophase and continues into its own step called cytokinesis. Some will tell you that cytokinesis is its own separate step after telophase has happened. It occurs completely after all of that. Point is, cytokinesis is the division of the cytoplasm. So this is when we have the cleavage furrow that actually pinches off. It kind of looks like a butt, which we'll look at. Okay, and it's when we actually have the two daughter cells pinch off to create two genetically identical daughter cells. They're just called daughter cells, not son cells. I couldn't tell you why. Sorry, boys. Right, but cytokinesis is a division of our cytoplasm, whereas we've already divided the nucleus and everything like that further in mitosis, okay? So here's what we're talking about. You have a contractile ring that's going to form at the cleavage furrow, and as this folds in, it starts to look like a butt. It's going to pinch off that cytoplasm and form two genetically identical daughter cells. So we talked about the control a little bit and how that's really important, and how all your cells can't divide at the same time or you'll die. And if your cells are going to be dividing too rapidly, that means that you can have tumors, which is just uncontrolled cell growth, things like that. So we have control of cellular division. We have go and stop signals that direct when a cell should do what, right? So our go signals are going to include critical surface area to volume ratios of the cells. So like we talked about this earlier, very large cells are very unhealthy. They need to reduce their size. They need to be smaller. They need a very high surface area to volume ratio, the smallest volume that they can have while maintaining a large surface area. Okay, this is important for our exchange with the external environment. If we're trying to bring in nutrients and we're trying to release waste products, if we have so much cytoplasm that you can't move anything in and out because it just takes forever, the cell's going to die. We need a very efficient transport system, and our membranes are really great at transporting things, but if we have huge cells that have a very large volume, that starts to become very difficult. We also have stop signals that are going to include the availability of space. Normal cells stop dividing when they come into contact with other cells. This is contact inhibition is what that's called. So our stop signals, contact inhibition. So when cells come into contact with other cells, they'll stop dividing. We have different proteins that help us control when one stage of the cell cycle stops and another begins. So two groups of proteins are crucial to the cell's ability to accomplish the S phase and enter mitosis. Remember that the S phase is synthesis, or the synthetic phase, which is when we're going to double the DNA. Why? Because mitosis, T for two, we're making two cells. We need two sets of DNA. So the DNA can boss around everything and let it know what to do in the new cells. We have cyclins that are regulatory proteins that accumulate during interphase. You have CDKs, which are cyclin-dependent kinases. So if they're dependent on cyclins, there better be a cyclin around. Am I right? Okay, so cyclin-dependent kinases are going to activate these cyclins when they bind to them. Then you have the cyclin that's bound to the CDK. It makes a complex. It just means that the two of them are attached. Don't let that scare you. Okay, and that is going to activate an enzyme cascade to help prepare for cellular division. Okay, our cyclins are destroyed after each mitotic cell division, and the process begins again. So you'll have a spike in our different cyclins as we're moving through the cell cycle, and then you will see that they get degraded, so then their specific levels will start to decrease as you are entering into the next stage. And this will just keep happening. You just build them and break them, and that's just a very normal process in order to help drive the cell through cellular division. You also have these guys called checkpoints. You saw them in the pictures. These checkpoints help to assist, like a check. That's specific there. It helps to make sure that everything is good to go before we make a copy. Because let's think about this. If you have something wrong with a cell and you're about to make a copy of that cell, now you have something wrong with two cells. And if it still doesn't go unchecked, if it's just like straight chilling, we're going to let you divide again. Now you're going to have four cells that have a mutation, if you will, some sort of change, something that's wrong with them. Okay, checkpoints are very important, because all of us aren't walking around with cancer from the second we're born to the second that we die, right? We have checkpoints that are key events in the cell cycle to help control and check what is happening. We can stop the cell or arrest the cell in these checkpoints to make sure that everything is fixed before we allow it to go through. You have a G1 checkpoint, you have a G2/M checkpoint. So this one, the G1 checkpoint, is a restriction point. It's the most important of the three major checkpoints here, because if a cell does not pass that G1 checkpoint, like that's a commitment step. Like once you pass that, you are going to divide, okay? Then it enters G0, in which no further division occurs.
Dynasty McClennan, please report to the Texas Tech House. Dynasty McClennan, reports to the Texas Tech House.
So checkpoints are also really important to help control how the cell cycle is going to progress. So we have a couple different checkpoints that are going to be like stopping points, like, hey, let me just check and make sure that everything's good to go before you keep going. Because what happens if a cell just keeps on going on through the cell cycle with just willy-nilly, no one's checking up on it, it's just doing its thing, dividing. What if it has something wrong with it? What if there's a DNA mutation? What if there's some proteins that aren't functioning correctly? What if we're just perpetuating that because we're letting it divide and divide and divide and divide? Suddenly we have a lot of cells that all have this problem. Well, yeah, that's a thing called cancer, right? That's bad. So we have these checkpoints that are in place in order to help us prevent things like that from happening. We have the G1 checkpoint, which is going to be the most important of the three major checkpoints. If a cell does not pass that particular checkpoint, it enters G0. G0 is pronounced G0, in which no further division is going to occur. So looking at this here, that's the G1 checkpoint. It's a restriction checkpoint. Then you have another one after S phase and G2 here. So this is the G2/M checkpoint. Some textbooks say that there's the one immediately after S. That's fine. You just need to know that basically the point of checkpoints is to make sure that everything has happened as it should, right? Because you don't want to double something that's a mistake. Some of you like to copy off of each other, and when you copy off of someone that, isn't doing so hot in the class, now you have two copies of the same mistake. And if this perpetuates, then suddenly the whole class is in trouble, right? Same thing for your body. You do not want to copy a mistake. You want to fix the mistake before a copy is made, so all of your cells are happy and healthy. Okay, so that's how we take one cell and turn it into two cells for the purposes of growth and repair. We have checkpoints, and we have those proteins, cyclins, and cyclin-dependent kinases that we talked about that help us push the cell through the cell cycle in order to create our two genetically identical daughter cells. Protein synthesis is going to be our next topic. That will be the last, hopefully, installment of our Chapter 3 notes for this unit. It's long, but thanks for bearing with me. Have a great day.